Hello, my name is Marina DiStefano, and I'm a clinical molecular genetics fellow at Harvard and a postdoc in the ClinGen resource. This is the final part of a 3-part training series on transcript annotation and curation. Part 3 will discuss a strategy for curating transcripts using the disease domain of hearing loss as an example. This is an outline of the entire series. In the first part, I covered why transcript curation matters, citing examples from the hearing loss clinical domain. I also provided an overview of transcript annotation bodies and their differences. In Part 2, I further discussed specific transcript curation resources. In this third part, you will hear about a specific transcript curation strategy that we deployed in the hearing loss domain. When a gene has multiple transcripts, how do you determine which transcript to use for variant annotation? It requires transcript curation, an example of which I'll detail now. This is the transcript curation process that we employed in the hearing loss domain. To read about this process in more detail, please refer to our paper in the Journal of Molecular Diagnostics. We took the 109 genes on the OTO genome, which is the hearing loss clinical testing panel at the Laboratory for Molecular Medicine, and separated them into 3 categories based on the number of transcripts per gene. Category 1 genes had 1 transcript and required less curation. Category 2 genes had multiple transcripts, but the longest transcript contained all unique exons. And Category 3 genes had multiple transcripts with unique exons and required thorough curation at the gene and variant level. Why are these categories important? They're important because of their impact on loss-of-function variant interpretation. If you're familiar with the ACMG guidelines, you know that PVS1, a loss-of-function variant in a gene where loss-of-function is a known mechanism of disease, is a strong piece of evidence for pathogenicity, very strong, in fact. Therefore, one must be sure that a variant results in true loss of function before applying this code. So, to put this in perspective in relation to the categories we have, in Category 1 genes, a loss of function variant would be true loss of function if it doesn't escape nonsense-mediated decay. For Categories 2 and 3, more curation must be done before a loss of function variant can be determined to be loss of function. Is the transcript of the loss of function variant biologically relevant? Is the exon where the predicted loss of function variant is located biologically relevant, or is it predicted to be spliced out? These are the categorization results. We were surprised to find that there was almost an even split of hearing loss genes across all of the categories. I suspect that this is not a disease area specific phenomenon, and that it further drives home the need for transcript curation. As expected, Category 3 genes harbored the most transcripts and the highest number of exons, as can be seen in the gray bars in the bar graph. As mentioned, Category 2 and 3 genes required even more curation, so I'll spend some time now discussing the exon-level curation we completed for Category 2 and 3 genes. For the C2 and C3 genes, we evaluated both gene-level data and exon-level data. For the gene-level analysis, we looked at the biological relevance of each transcript by looking at expression in the literature and GTEx. Refer to Video 2 for an in-depth explanation of how to use both of these resources. I should point out that GTEx does not have our tissue of interest, cochlea, so we had to examine broader expression patterns. For exon-level variation, we looked at high-frequency, high-quality loss-of-function variants in NOMAD. We used both sets of information to classify exons into the buckets you see in the table on the right. A clinically significant exon had literature support for its relevance, and/or no predicted alternative splicing in GTEx. An exon of uncertain significance did not have any expression data in the literature and had predicted alternative splicing in GTEx, or one high-frequency loss-of-function variant in NOMAD. We defined greater than or equal to 0.3% as high frequency because it is our likely benign cutoff for hearing loss variant curation. A clinically insignificant exon was confirmed as insignificant in the literature and/or had multiple high-frequency loss-of-function variants in NOMAD. Non-coding exons are self-explanatory. Once we classified the exons, we then evaluated interpreted variation by pulling all disease-causing mutations from the human gene mutation database and all likely pathogenic and pathogenic variants from ClinVar, from submitters with assertion criteria. These are the classification results here in the bar graph on the right. As you can see, each category harbored exons of uncertain significance, with C3 harboring the most. However, only C1 and C3 transcripts had clinically insignificant exons. I want to detail an example of how we classified exons as uncertain significance now. Endothelin 3 is a gene that is associated with Waardenburg syndrome, a syndrome with hearing loss and pigment abnormalities. RefSeq has 5 curated NM transcripts. Refer to video 1 for an explanation of RefSeq transcripts. All of these transcripts share exons 1 to 3 and 5, with coding exons being listed in dark blue and non-coding in light blue. However, exon 4, which is boxed in red, is only present in 3 of the transcripts. When you look in Nomad, there is a frameshift variant that is present in 0.6% of Finnish European alleles in Nomad, including 2 homozygotes. Additionally, when you look at this exon in GTEx, it is predicted to be spliced out in tissues where endothelin 3 is expressed. Therefore, we classified exon 4 as an exon of uncertain significance. This calls into question the pathogenicity of the 2 DM variants from HDMD that are located in this exon and points out that they need further curation. When we pull all of the DM variants from HDMD and the likely pathogenic and pathogenic variants from ClinVar for the exons of uncertain significance and the clinically insignificant exons, we can see that many variants require further evaluation. 6% of exons were classified as uncertain significance. 124 variants that were DM or likely pathogenic or pathogenic fell in these exons. I just want to end with a brief discussion about technically challenging regions. Although it is not a curator's job to know about all of the technical challenges of a clinical pipeline, it may often be critical to be aware of known sequencing issues with certain genes. These can be specified by disease experts and might be part of the pre-curation process. If they are particularly common problems, they might even be alluded to in published literature. What we did to evaluate technically challenging regions was take the next-gen sequencing data from 2 clinical labs and evaluate average mapping quality and depth of coverage for the 109 hearing loss genes on our clinical panel. When we performed this analysis, we found 43 technically challenging exons in 20 different genes that might lead to false variant calls. In this case, these regions might be ones where we might be cautious scoring variants if they aren't confirmed by orthogonal methods. One very well-known example in the hearing loss field is stereocylin. This gene has high homology to a pseudogene, which can be seen when you plot the depth of coverage and mapping quality. They are quite low from exons 1 to 13 or so, indicating that variants in this region should be scored or evaluated with caution. Special care should be placed on reading the methods of papers to determine if variants in this region were detected another way before they are scored. Because it requires substantial manual curation, scaling transcript curation is a challenge. We have started discussions of scaling and can likely automate some parts of the process, such as auto-categorizing genes based on RefSeq transcripts and filtering out exons by using high-frequency loss-of-function variants or GTEx expression. Unfortunately, literature curation is still a manual process at this time, and we are limited by what GTEx contains as far as relevant tissues and time points. In conclusion, I'm hoping through this transcript curation series, I've convinced you that transcript curation is critical for variant interpretation, and have provided you with the resources to curate transcripts in your disease area of interest. I'd like to take this time to acknowledge all of those individuals who worked with me on the transcript curation project, and thank you for listening.